See, we have a practical aspect in our UG as well as PG classes, and this particular practical exercise is related to the field of evolutionary biology. In this particular practical exercise, we demonstrate the phenomenon of natural selection by making cross between wild type flies, that is red-eyed flies, and mutant flies, which are actually white-eyed flies of Drosophila melanogaster, and this we perform under laboratory conditions. So it is an experiment which is demonstrated to all the students of UG and PG classes, and they really enjoy this subject because it gives very good idea that how natural selection operates against certain types and it favors some other types. So before I explain this specific experiment, we should be acquainted that natural selection actually deals with the differential survival and reproduction of different phenotypes or different genotypes. That is, in the population, individuals of different phenotypes or they will be genetically different from each other, and all such individuals will not have equal survival value. They will definitely be varying in their survival rate as well as reproductive ability. So, and this aspect we have already studied in our theory classes by considering the case of industrial melanism, which was studied in case of peppered moth, that is Biston betularia, and the aspect of natural selection can be studied in case of bacteria, that is also explained in a number of books by taking example of antibiotics resistance in bacteria, and this phenomenon has also been studied in insects by considering insecticide resistance in insect population. But here the experiment which I am going to demonstrate is related to Drosophila melanogaster, wild type and mutant types. So you can have the complete idea about this experiment. Let me explain this particular topic. See, we actually use Drosophila flies. We need wild type flies which are already cultured in our laboratories. So what we take actually virgin flies. Like we take red-eyed females. These are collected when they come out from the pupal case. So red-eyed females are collected. Then white-eyed females, they are collected from culture bottles. We actually have these two different kinds of flies being reared in the laboratory. We have stocks of Drosophila melanogaster, which are totally normal in their phenotype, and then we have Drosophila flies which are actually white-eyed. So these two types of stocks are present in the laboratory and they are being reared for several years. So we collect virgin red-eyed females, white-eyed females, then red-eyed males and white-eyed males. And to start an experiment, we actually take 5 flies of each type, that is 5 red-eyed females, 5 white-eyed females, then 5 red-eyed males and 5 white-eyed males. So we can put these 20 flies in a food bottle. Okay, so to start an experiment, we actually need 20 flies and we will be putting these 20 flies in a food bottle. If we have several students in the class, so many students are there, in that case we will make batches and each batch may contain 5 or 6 students, and that batch will be provided a food bottle. They will be putting 20 flies in that food bottle. So if 10 batches are there in the class, then each batch will be starting experiment this way by keeping 20 flies in the food bottle. What they will do, that they will allow these flies to remain in the food bottle for 5 days, because we hope that in these 5 days there will be mating among the males and females of these flies, and then after 5 days we actually remove all these flies, we discard these flies. So we are able to get the next generation flies, that is F1 flies, in about 12 days. So after 12 or 13 days, or maybe after 14 days, you can observe the F1 progeny. So let me show what will be the result of this experiment and how this experiment will be continued. As I told, we actually need 20 flies to start an experiment, and these are actually parental flies. I am referring them as parental flies. So as I told, these flies will be put in the food bottles. They will remain in the bottle for 5 days. After 5 days, they will be discarded, and we hope that in these 5 days there will be enough egg laying, and then the F1 progeny will be available. So you have to count the F1 progeny with respect to their sex as well as phenotype. So what we find, that suppose I have shown here a data, means such kind of experiments have already been done, and based on the available data I am showing this entire experiment. Suppose in F1 you observe 455 flies being red-eyed females. So these are females and they are red-eyed. You observe 363 flies which are females but they are white-eyed. Then 460 flies are there. These are males and red-eyed males. And 390 flies are there which are white-eyed males. So as I just told, that you have to count all the flies of this F1 generation with respect to their sex, whether they are male or female, and what is their phenotype, whether they are red-eyed or white-eyed. And after counting them, after counting all these flies, you will have to transfer them in the food bottles. All these flies, you will not be sorting out any one of them. You have to carefully perform this experiment so that F1 flies could be obtained, and these F1 flies, after counting, they will be pooled with each other, means they will be actually kept together in the food bottle, so that you can get F2 flies, and you will keep them in the food bottle just for 2 or 3 days, because since they are very large in number, means they are quite good in number, they will be able to produce progeny just in means they will be able to mate and produce progeny. So just to avoid overcrowding, you have to allow them in the food bottle just for 2 or 3 days, and after that you will be able to get F2 flies in the next 13-14 days. So again these F2 flies will be observed for their phenotype as well as for their sex. So what you are doing, in every generation you are counting the number of males and females, whether they are red-eyed or white-eyed, and then after counting they will be transferred in fresh food bottles. They will be allowed in the bottles for 2 days, and after that they will be discarded, and their next generation progeny will be observed. So this you will go on doing for as many generations as you can, and suppose this experiment is continued for 7 generations. Up to 7 generations we have actually uh, continued this, or we did this experiment. So what we are observing here, that we are getting these flies, the red-eyed females are available, the white-eyed females are also being observed in these generations, but we are observing that the white-eyed females, their number is going down. If you carefully observe this data, you find that the number of white-eyed females, that is going down. And you will also observe that the white-eyed males, their number will also be going down. But red-eyed males, they are not actually declining. They are almost equal to the number of females, that is red-eyed females. So red-eyed females and red-eyed males, they are not being affected much more number-wise, but mutant males and females, their number is going down in every generation. Now, if we continue this experiment for some more generation, then we will find that the mutant types, that is mutant females and males, they will go on dwindling, they will go on decreasing in number, and maybe in 25 generations or 26 generations, you will not be getting any mutant fly in the culture bottle, because people have done this kind of experiment. They have continued this experiment for 26 or 27 generations, and by that time not a single mutant fly will be available. So why this happens, that I will discuss later, but you should know that if you continue this experiment for further generation, you will find further decrease in the number of mutant flies. So definitely something would be acting in this population, and that is why it is happening. But important point I have to say is that we will be using this male data for the analysis purpose. This male data regarding red-eyed males and white-eyed males, this will be of much use. Why this will be of much use? This you can understand by the next presentation. What I'm saying, that we will be considering only red-eyed males and white-eyed males data. Why? Because we are knowing the genotype of these two types. We know that this white eye feature is a sex-linked character. Actually, the gene deciding this particular feature is situated on the X chromosome. So it is a sex-linked feature. And those males which are red-eyed, genotypically they are actually W plus, means plus allele is the normal allele. So it is present on the X chromosome. This is small chromosome is Y chromosome. So these males are actually XY, and there is presence of normal allele here. Those males which are white-eyed, genotypically means they have recessive allele here on the X chromosome, and this one is Y chromosome. So when we observe a male with a red eye, we immediately come to know about the genotype of this individual. Likewise, if there is a white-eyed male, we know the genotype of this fellow. And if we have a female which is white-eyed, we can also know the genotype of this female. We know that on both of her X chromosome, the recessive allele will be situated. But we do not know the genotype of red-eyed females. So, because red-eyed female will be having, definitely the normal allele, but such females may be heterozygous also. So, if a female is red-eyed, its genotype may be W plus, W plus, or W plus and W. So she may be homozygous or she may be carrier. So that is why the females, particularly red-eyed females, their genotype could not be known. So we cannot actually and even if you have, it means if you continue this experiment for 20 generations, and if you find females only, in such case there may be one or two female which may be carrier. So considering that the genotype of females, particularly red-eyed females, could not be known, we are going to consider only the male individuals in this case, because at least the genotype of male flies could be known. So we are going to utilize the data of this male population, and what we find, this data is exactly the same one which I just showed. So we are considering the red-eyed males and females of this parental population. In the parental case, 5 males and 5 white-eyed and red-eyed males were taken, and then F1, F2, F3 up to F7, the number of red-eyed males and white-eyed males, that is given in this table. Now what we can do, we can count the number of male individuals of a particular generation. Like F1 generation, 460 red-eyed males and 390 white-eyed males are there. So we are going to take total of these two, and it has come 850. So out of 850, 390 are mutant types. So when we calculate its percentage, it is coming 45.88. So this is percentage of flies which are white-eyed. Another point is that in the parental population we took 5 males red-eyed and 5 males white-eyed. So here you can say that out of 10 there are 5 white-eyed males, so its frequency is 50%, or you can say 0.5. So 50% males are white-eyed. The frequency of this W allele is 50% here. Then as I just said, in the F1 generation you have observed that the frequency of this or percentage of W allele is 45.88. In F2 again you will count the number 525 plus 210, it comes 735. So 210 divided by 735, and when you will multiply it by 100 to know the percentage, it will come 28.57. So this way you can calculate the percentage of this white eye allele in the population, and that percentage you are observing here. In the parental case it was 50%, in the next generation it became 45.88, in F2 generation it became 28.57, then 19.67, then 14%. So we are observing a steady, a continuous decrease in the frequency of this white eye allele. And as I said, if you continue this experiment for some more generation, you will find further decrease in the frequency of this W allele. And based on this data, based on the data of this W allele, you can make a graph. So this is a graph that is showing frequency of recessive allele in the different generations of Drosophila melanogaster. So, and this frequency is shown in percentage. So this is percent of W allele, and actually this 1 means parental generation. In 1 means it is actually parental generation, you can say generation 0. The frequency of this W allele was 50%. Then in further generations it went down, and up to 7th generation its frequency has come close to this 10%. So there is continuous decrease in the frequency of white eye allele. Now the question is that why there is a steady decline, continuous decline in the number of white-eyed flies in the culture bottle. We want to know this, that why this kind of trend is there. And people have done experiments, people have done behavior related experiments. They have observed actually mating success of red-eyed and white-eyed males, and they have seen receptivity of red-eyed and white-eyed females. They have also observed the fecundity and productivity of white-eyed and red-eyed females. Based on all such analysis, I am citing here 3 major points. The first point of this decline in the mutant type is the red-eyed males are more successful in mating than white-eyed males. So this is first point, that is red-eyed males, they are more prompt, they are more active in mating than white-eyed males. So this is first reason. Second one is red-eyed as well as white-eyed females prefer to mate with red-eyed males. This is another reason, that both kind of females, that is mutant as well as wild type, they prefer to mate with the males of red-eyed type. And the fecundity and productivity of red-eyed females are more than white-eyed females. So as a result of this, the number of red-eyed flies, that is red-eyed females as well as red-eyed males, that go on increasing in further generations, and we observe continuous decline in the number of mutant flies. So this is one means aspect. You can see how natural selection operates, because these are the factors. These are the three major reasons which are hidden reasons which actually cause the decline of mutant flies. And a time comes when you will not be able to observe even a single mutant fly in the population. So this is a very important kind of experiment which we demonstrate, and which you can understand by going through this presentation.